Hi friends, today we are going to see the past year NEET questions for the chapter Cell, the Unit of Life, and today in this video we will cover part 1. Now let's begin with the first question. Read it here. This question was asked in the year 2015. Identify the correct order of organization of genetic material from the largest to the smallest. Okay, so you're given answers here, you're given 4 options here. Before we go into the 4 options, let us actually discuss each keyword that is given in the option. Now let's take genome first. So what is genome? How can you describe genome? Genome is the total genetic material of an individual. Okay, it is you're talking about the total genetic material here. Okay, now what is chromosome? It is a packed and organized structure containing most of the DNA of the living organism. So it's a packed and organized structure which contains most of the DNA of the living organism. Okay, most, predominantly. Now coming to the next keyword given here, nucleotide. What is nucleotide? It is one of the structural component, or you can call this as a building block of DNA or RNA. As you all know, a nucleotide will be made up of 3 components: a sugar, a base, all that. Likewise, now what is gene? It is a segment of a DNA that encodes for a protein. Now since we have completed all the 4 keywords, now it is very important to arrange them from the ascending, from the largest to the smallest order. Which weighs or which is huge, which is large. So the correct order is genome, okay, chromosome, gene and nucleotide. We have arranged it from the largest to the smallest. Okay, therefore the correct answer is option B, genome, chromosome, gene and nucleotide. Okay, so now let's move to the next question. This question was asked in the year 2015. A protoplast is a cell. Okay, it's a dash. Okay, what is protoplast? Have you ever thought what a protoplast is, or have you ever heard about what is protoplast? A protoplast is the protoplasm. You can call it anyway. You can call it protoplast or protoplasm. Okay, now this protoplast is actually, or it is seen in plants or bacterial cells where the cell wall has been removed. The cell wall has been removed. Okay, they are cells which have had their cell wall removed, usually by digestion enzymes. Some of the enzymes are cellulase. Okay, one of the enzyme is cellulase. You have many more enzymes. So once the cell wall is removed, the resulting protoplast will be spherical in shape. Okay, so a protoplast is a cell without a cell wall. Okay, cell wall is absent for protoplast. Therefore the correct answer is option C, without cell wall. As I already said, it will look spherical once the cell wall is being removed. Actually it will get digested by the enzymes that are present inside the plant or the bacterial cell. That is why the cell wall gets digested and it looks spherical. So this is how, to your right side, what image you see is, this is how they will you can see them under a microscope. Okay, therefore the correct answer to this question is option C, without a cell wall. Now let's move to the next question here. Look at this question here. A major breakthrough in the studies of cells came with the development of electron microscope, and this is because this question was asked in the year 2006. Now we all know that after people had discovered something called microorganisms which cannot be seen through your naked eyes, we all understand, we all know so much about them is because of the studies that we carry out with the help of a microscope. Of course, as I already mentioned, microorganisms, they cannot be seen physically, but only when you collect them and when you make a sphere under a microscope, you can view them. So a major breakthrough in the studies of the cells came with the development of electron microscope. Now this electron microscope what is the importance? What actually happened, or what major breakthrough it brought? If you see, as you all know, a microscope with high magnification and resolution employing electron beams of light, okay, employing electron beams of light in the place using electron lenses, it will actually accelerate, and the image that has to be viewed will be viewed clearly. So this electron microscope, if you see, it uses beam of acceleration of electrons as a source of illumination, so that we can see the ultrastructure of many subcellular or cell organelles. Okay, therefore the correct answer to this question is option D. The resolution power of the electron microscope is much higher than the light microscope. Light microscope is the ones that we use in all schools and colleges. A normal study, you don't have, where you just have to go through the cell wall and the external parts, viewing of an algae or a fungi. But this electron microscope, you will be able to even view the cellular organelles, so that much high it has the magnification power. Okay, so magnifying is to elaborate things and show, right? A major breakthrough in the cell studies came with the development of the electron microscope, which has a greater resolution power when compared to the light microscope. Therefore the correct answer to this question is option D. Now let's move to the next question here. Now this question was asked again in the year 2005. Let's read it here. A student wishes to study the cell structure under the light microscope, which has about 10x eyepiece and a 45x objective. Okay, he should illuminate the object by which one of the following colors of light, so that the best possible resolution that the student can view in. Here, if you see, before we go into the answer, okay, what do you mean by resolution power? Why do you need a perfect resolution to view something which has to be viewed on a microscope? Resolving power or the resolution power? Okay, you can call it resolution, or you can call it as resolving power. Okay, resolving power. So this is the ability of the lens to distinguish fine details and structure. Okay, even the things that we leave without noticing, it is very important that when this resolving power is kept perfect, you can even find some fine things under them. Resolving power depends upon two factors. What are the two factors? One is wavelength of the light used for illumination, and the other one is the power of the objective lens. Okay, it is dependent upon two factors. Okay, it is dependent upon two factors. Now you have four options here: blue, yellow, green and red. Okay, therefore to your right side you can see a small calculation here. Resolving power or limit of resolution is the ability of the eye to distinguish two close objects as separate. Though they are placed at a very close distance, your eyes should get that tendency to distinguish the two close objects. So for that you will use B's equation. Okay, the equation is given here. L to the base m, okay, is equal to 0.61 lambda divided by Na. Na stands for numerical aperture. Okay, and lambda is the wavelength of light. Since we have this equation, I'll tell you the answer. The power of objective lens, okay, resolving power and the wavelength of light, as we all know, we have a formula here. Since the limit of the resolving power of a microscope is supposed to be fixed by the structure of light, shorter the wavelength, higher the clarity. You have to remember this. Whenever you use a microscope, if you use something with a shorter wavelength, your objects will be viewed clearly. Okay, so shorter the wavelength, I have written it somewhere. No. Okay, shorter the wavelength of a visible light, okay, it will give the maximum resolution. Okay, shortest. Okay, this is very important. So this will give, when the wavelength is very short, it gives you a maximum resolution. Among yellow, green, red and blue light, okay, blue which has about 500 nanometer. when compared to the other colors. Blue has the shortest wavelength, and therefore it will give you the best resolution. Therefore the best resolution for that person, for that student to view whatever is to be viewed, the correct answer is option A, blue. Now you understand why it is important to use a shorter wavelength to have high resolution power or resolving power. Okay, blue has only 500 nanometer. Okay, now let's move to the next question here. This question was asked in the year 1993. Names of scaladin, squan are associated with dash. Scaladin. Okay, now here if you see, this question was asked in the year 1993. You have 4 options here, right? Okay, now as we all know, I think by now you have guessed the answer, as scaladin and squan are associated with, they are not associated with protoplasm, they are not associated with theory, they are associated with theory but not with the theory of cell lineage. The correct answer goes to option B, cell theory. Okay, cell theory, it was proposed by scaladin and squan in the year 1839, according to which all living organisms are composed of cells. Cell theory is also known as cell principle or cell doctrine. Okay, therefore the correct answer to this question is option B. This question you don't have to, I think you don't have, you don't need any much explanation, because it's just they are just asking us these 2 people are responsible for what. As we all know the answer, the correct answer goes to cell theory. Okay, now let's move to the next question here. This question seems to be big, don't get scared. This question was asked in the year 1993. Okay, let's read it. Which is correct about cell theory? So last question we saw who actually is associated with cell theory. Now we are going to see which one is correct about cell theory in view of current status of our knowledge about cell structure. So they are comparing. We are talking about the current status, right? We are talking about the current status here. Okay, the exceptions to the cell theory are viruses. Okay, are viruses. Look at the first option. It needs modification due to the discovery of subcellular structures like chloroplast and mitochondria. See, this discovery is very old. Okay, we're talking about the current status, so this one is not the answer. Look at the second option here. Modified cell theory means that living beings are composed of cells and they're capable of reproducing. This is also something old. We're all talking about the current status. Now look at the third one. Cell theory does not hold any good because of all living beings, especially viruses which do not have a cellular organization. Now this is the right answer. As we all know, virus neither belongs to prokaryotes or eukaryotes, right? They possess a cellular machinery which sometimes contains DNA, which sometimes does not, which sometimes contains an RNA. So when there is no proper cellular organization, it is very hard for us to classify them and to tell them this is what they are, right? They consist of a DNA or an RNA core which surrounds the protein sheath, and they also lack cellular organization. Viruses are usually acellular. Do you agree with me? Why do you call them acellular? Acellular means they lack the most of the components of the cells. When you say components of the cell, you can talk about the organelles like ribosomes, mitochondria, plasma membrane. These kind of things are absolutely absent in viruses. So the correct answer to this question is option C. Cell theory does not hold good because all living beings, especially viruses, they do not have a proper cellular organization, and therefore when it comes to cell theory, we cannot classify them and tell them this is what they are correctly. Okay, therefore the correct answer to this question is option C, cell theory. Okay, so viruses is what is a problem now when it comes to current status, since we cannot classify them correctly. Okay, now let's move to the next question here. This question was asked in the year 1992. This is a very easy question. In fact, angstrom is equal to dash. I think by now all of you would have guessed the answer. Yes, the right answer is option C. Okay, now angstrom, so this is a person, he's called angstrom. He was the one who discovered this method. And if you see, angstrom is equal to 0.001 micrometer. And again, you have 1 angstrom is equal to 10 to the power of minus 10 meter, which is equal to 10 to the power of minus 8 centimeter. So these are just calculations. Now, what is this angstrom actually? It is a unit of length. Okay, this is a unit of length. Usually, this angstrom, if you see, it's actually a Swedish alphabet. It is a Swedish alphabet. Okay, and this was the person who discovered this particular unit called angstrom. Now this angstrom, a very important thing to note here is angstrom doesn't come under SI unit. They are not under SI unit, but they are under metric system. Okay, they are under metric system. Therefore, the correct answer to this question is option C. I've given you extra information about angstrom also. So now let's move to the next question here. Now look at this question. It was asked in the year 1992. Again, it's about microscope. Electron microscope has high resolution power, and this is due to dash. Okay, we all know, we already saw in the earlier slides, why is it that we have high magnification and it is more it has more resolving power than a light microscope. We saw in the earlier slides. So why is it having that? I think we all by now we have guessed the answer, because in the earlier part, in the earlier slide, I mean in the earlier question, we saw that electron microscope has high resolution power than a light microscope because of the short wavelength, right? Shorter the wavelength, the resolving power will be high. Therefore, the correct answer to this question is very low wavelength of the electron beam. Okay, very low wavelength of the electron beam. Resolution is the power of a microscope to distinguish between two particles that are closely situated, and that distinguish will happen in your eyes. The eyes has a capacity to distinguish between two close objects, but that two close objects, you need a specific color. We already saw in the earlier part about blue color. Blue color, which has the shortest wavelength, will give you the highest resolution. Therefore, the electron microscope works on this particular theme, where it uses shorter wavelength to experience and give you a high resolution and more clearer image. Okay, therefore the correct answer to this question is option B. Now let's move to the next question. This question was asked in 1991. Resolution power is the ability of dash. Right? Resolution power is the ability to dash. Now, when we view something under microscope, it can be a microorganism or it can be any organelle, anything. It is very important that we distinguish, right? Now, for example, I'll tell you an example. Imagine we are distinguishing bacteria. Okay, bacteria are of two types. I mean, they are of many types, but here we talk about the staining method, gram positive and gram negative. Now you can ask me, what is this? See, these are the gram positive, gram negative. It's nothing but the stains that the bacteria will take. If it is a gram positive bacteria, it will take the purple stain, and if it is gram negative, it will take a pink stain. So this is how they differentiate between gram positive and gram negative bacteria. Now, the same smear that you prepare and you keep it under the microscope will have a combination of everything, but still when you happen to view it under microscope, you are able to distinguish between the purple and the pink. The color variation is there, but this variation, this distinguishing character is happening because of the wavelength that the microscope will absorb from the visible light. Therefore, resolution power is the ability to distinguish between two close objects. Okay, the ability to distinguish two close objects. Therefore, the correct answer to this question is option B, distinguish two close objects. Okay, now let's go to the next question. Now, this question was asked in the year 1990. Magnification, okay, magnification of a compound microscope is not connected with dash. Okay, fine. Now, magnification is to elaborate something and view it clearly. You go in depth. The meaning of magnification is only that. Magnification of a compound microscope is not connected with dash. Magnification of compound microscope, it depends upon various things, but it is not connected to one particular thing, and that is called as numerical aperture. Why? See, the magnifying power is represented by the symbol x. We all say no, 10x, 45x. So it is always denoted with the help of x. The total magnification of a microscope is obtained by multiplying the objective magnification. So you're multiplying objective magnification and ocular lens magnification. Example, if the magnifying power of an ocular lens is 10x, imagine you have 10x and that of the objective is 40x. Okay, then the total magnifying power of the microscope is 10x into 40x, which is equal to, so whatever it is, you will view it in this way. Okay, therefore the correct answer will be 40x. Magnification power, magnification of a compound microscope is the power of enlargement. As I already said, you will elaborate things and you will view it under the microscope. Magnification of a compound microscope is a product of magnification of the objective lens and the ocular lens. I gave you an example also. Okay, you will multiply both and you will get the exact view of the microscope, of a compound microscope. Therefore, the correct answer to this question is option. See, let me tell you, when I told that calculation of 10 into 40, it gives you 400 times. It is 400. I'll explain you. 10 into 40. I told you it is the multiplication of the multiplying the objective magnification and the ocular lens magnification. So 10x is the magnification, this thing, and the ocular lens is 40x. So this becomes 400. So imagine the magnification will be 400 times. Okay, therefore magnification of the compound microscope is not connected with numerical aperture. Okay, option A is the right answer. Now let's move to the next question here. This question was asked in the year 1989. Organelles can be separated from cell homogenate through dash. Okay. Chromatography is to separate proteins. X-ray, you know, it doesn't separate anything. Autoradiography also is the same thing. Therefore, the correct answer is differential centrifugation. Now, what do you mean by centrifuge? How does the principle work for a centrifuge? Centrifuge is an instrument that is used to create, or it separates components that are present in the mixture with the help of the centrifugal force. The homogenate of the cell organelles is allowed to undergo centrifugation, which will allow to centrifuge, and it will get separated into supernatant and pellet. The pellet and the supernatant. This process of centrifugation, you call it as differential centrifugation. It is also called as differential velocity centrifugation, and this process, if you see, it is commonly seen in biochemistry, okay, and biology. They use highly to separate these cell organelles, and they will do many experiments. Okay, therefore the correct answer to this question is organelles can be separated from cell homogenate is through differential centrifugation. So to your right side you can see a diagram which shows the various organelles that have been separated with the help of the differential centrifugation. See, at each time, at first thing you got the nuclei separated, then you have mitochondria separated, lysosomes, microsomes, cytosol, all these got separated with the help of the differential centrifugation. Based on the centrifugal force they work. Okay, therefore the correct answer to this question is option C. Now let's move to the next question here. Now this question was asked in the year 2015. Which one of the following is not an inclusion body found in prokaryotes? Okay, inclusion bodies are commonly present in prokaryotes. but this one is not present in prokaryote. The correct answer goes to polysome. What is polysome? Polysome is not found in prokaryotes, but this is found in eukaryotes. Okay, this inclusion body is found in eukaryotes. Okay, these are cluster of ribosomes that are held together by a strand of messenger RNA, mRNA you will call it as, right? You would have heard mRNA or messenger RNA, which actually is important for translating. During translation, I think we all know there's some process called transcription translation that happens in eukaryotes. So this polysome is very important during the translating purpose or translation process. Therefore, the inclusion body that is not found in prokaryotes is polysome. Okay, option C is a right answer. Now let's move to the next question here. Cell wall is absent in dash. Again asked in the year 2015. Okay, we all know Aspergillus, Funaria, Nostoc. These are all algae, blue green algae. They all have a cell wall. Okay, the only thing left out is Mycoplasma. Mycoplasma belongs to the genus of bacteria that lacks cell wall around their cell membrane. Okay, so due to the absence of cell membrane, they become resistant. Why they are resistant? And to what they are resistant? Did you ever think about that? See, cell wall makes a bacteria more resistant, but we have heard that cell wall gives protection for the animals or for the microorganisms, but here it's just opposite. It gives resistance, it seems. Have you ever thought about it? I'll tell you why. If you see when we use antibiotics for any kind of disorder that happens in your body, antibiotics are designed in a way that these will go attack the cell wall of the microorganism, thereby killing the microorganism. Now these mycoplasma, sometimes they are parasitic, that is, they might become pathogenic also when they cause diseases in humans and animals. But here if you see, the antibiotics will not work because they do not have cell wall. So what happens? The antibiotics will go and attack cell wall. Since they don't have cell wall, the antibiotics work will become nil, and therefore it becomes resistant and thereby attacks the human or the animals that it is under control. Therefore, Mycoplasma is resistant because of cell wall. I think I'm clear with what I said. Therefore, I'll give you an example. Sometimes they will use beta-lactam antibiotics. So this will not work for Mycoplasma and all, since they don't have a cell wall. Therefore, the correct answer to this question is Mycoplasma lacks cell wall. Okay, can you understand the difference? Since they don't have cell wall, the antibiotics cannot attack the target. Therefore, Mycoplasma becomes resistant. Therefore, the correct answer is option B. Look at the next question here. Which structure perform the function of mitochondria in bacteria? Okay, they are asking which one will function as mitochondria in bacteria, and this question was asked in the year 2014. Fine. You have many, you are given 4 options: ribosomes, cell wall, nucleoid, mesosomes. Each function is unique. So out of the 4, the correct answer comes to mesosomes. What is mesosomes? Mesosomes are organelles of bacteria that appear as an invagination or invagination, you can call it as any way around, invagination or invagination of the plasma membrane, and they actually they function either in DNA replication. What is their function? If you ask me, they either function in DNA replication, okay, or they will help in cell division, and also sometimes in the excretion of enzymes, exoenzymes. Therefore, the correct answer to this question is mesosomes. Okay, in some bacteria like Bacillus subtilis, this is a gram-positive bacteria. Okay, Bacillus will be rod-shaped bacteria like this. So this is how it will appear under a microscope. So these are actually performing the function of mitochondria. Since they don't have mitochondria, mesosome will take that activity of mitochondria and help the bacteria to function. Therefore, the correct answer is option D. Now let's move to the next question here. The term glycolax is used for dash. Okay, you have 4 options here. Now before that, you should know what is glycolax, right? This is actually the outermost coating of the bacterial cell. So what is it called as? It is called the outermost coat of the bacterial cell or the cell wall, which is rich in polysaccharides. Okay, they are rich in polysaccharides. They are sugars. Polysaccharides are long chain of sugars, right? The other name for glycolax is mucilage. Have you ever heard about this? Mucilage. Okay, mucilage means they are viscous secretion, or you can call it as viscous body fluid. Okay, this is actually called the capsule, which gives the gummy sticky nature. What is the function, if you see, of glycolax? It protects the cell from desiccation. Desiccation means when the bacterial cells are prone to heat, okay, they might get destroyed. But this glycolax will prevent the cell from getting destroyed through desiccation or some toxins, and it also prevents the attachment of any foreign material over the surface. Therefore, the correct answer to this question is option A. It is a layer that is surrounding the cell wall of the bacteria, which is an extra covering that gives the bacteria more protection, especially from desiccation, toxins, and any foreign material, so that it doesn't get adhered to the bacteria and destroy the bacteria. So this is actually also called as mucilage, which means viscous fluid, sticky in nature. Okay, fine. So with this, we complete the part 1, cell, the unit of life. We all will meet in part 2. Thank you.